All right, welcome back. So this week, moving on to the immune system, as you can see. So I don't know about you, but I've learned a lot more about the immune system during 2020 and 2021 than I ever knew before. So hopefully, this will make some of the stuff we talked about back with that first module where we covered chapter 3. We talked about cells, and we talked about mRNA, and we talked about the COVID vaccines. There was a lot in those articles where it talked about antigen presenting cells and helper T cells and B cells and cytotoxic T cells and all that kind of stuff. And that probably didn't make a ton of sense to you. I think that the articles did a pretty good job of explaining those things in layman's terms, but my hope is that by the end of this discussion, by the end of this chapter, all of that stuff will make more sense. You'll have an idea of what an antigen presenting cell is and why that matters, what a T cell is versus a B cell, what they do, and then how that affects your immune response. So in talking about the immune system, some of the terminology that you've probably seen related to the immune system discusses the fact or the common division of the immune system into two broad branches. The first is the innate or non-specific immunity. So that is basically just helping us to keep all pathogens at bay. And then the specific branch, or the adaptive branch of the immune system, attacks a particular pathogen. So you can see the definition of immunity there. So it's the ability of the body to resist agents that cause disease. So those agents that cause disease, of course, could be things like bacteria, viruses, funguses, or fungi, all kinds of different things like that. So the immunity then is our ability to repel them because, as you would expect, we are constantly under attack. You've got bacteria that are constantly trying to get in and would cause infections if they did. And when I say trying to get into your body, viruses that are trying to get in and cause infections if they did, all those kinds of things, right? So one important term to probably get out of the way at the beginning, or two topics to discuss here at the beginning, I guess. Let's go with terms. So there's antigens and antibodies. So we've seen a lot about antigens. So antigens, and you'll see this again on a later slide, but just at the outset, an antigen is something that is going to provoke an immune response. So for example, we have to get COVID tested at the moment that I'm recording this. Students have to get tested once a week. Faculty have to get tested every other week. But when you click on that link, it says it's COVID antigen testing. And so what that testing looks for is part of the virus that would provoke the immune response, is looking for that particular antigen. So an antigen then is something that would generate an immune response. An antibody, on the other hand, is something that is generated by our immune system, specifically by B cells, and we'll talk about what those are, but specifically they're generated by B cells, and antibodies help mark antigens for destruction. So you have cells that are phagocytes, which kill other cells by eating them, basically by engulfing them. And so one of the things that antibodies do is they attach to the antigen, the pathogen, if you will, right? So the antibody attaches to the antigen, and then that's going to mark that particular antigen for destruction. It provides a handle, if you will, on that antigen that makes it easier for those phagocytes to hang on to that particular pathogen. And here I'm using pathogen, antigen interchangeably because they, for all intents and purposes now, are the same. So that's what an antigen is. So an antigen, the thing to know, it provokes an immune response. The antibodies are part of our immune response where we mark that foreign particle or we mark that foreign cell for destruction. So that's what antibodies do. They help mark those cells for destruction. And we'll talk about some other things that antibodies do. But in fact, the term antigen is a contraction. It means antibody generating, right? And so whichever particular thing, so whether it is part of the COVID virus, the antigen aspect of it generates antibodies or causes our B cells to generate antibodies to mark it for destruction. So with respect to the innate or non-specific immunity, so you can see on the slide here, our innate defenses. So basically, the way this chapter is laid out in both this textbook and others that I've used is that the immune system is kind of broken into 3 lines of defense. And so the first line of defense is part of our innate defenses, and so those are our surface barriers. So the first line of defense is effectively keeping whatever the pathogen is out. So that's kind of where you see a lot of the stuff about, wash your hands and all of the hand sanitizer that's everywhere. The idea is to augment those surface barriers because, the COVID-19 virus can't move through your skin. But if you, let's say, were to have it on your hands and then somehow, touch inside of your mouth or something like that, touch food that then goes in your mouth, then it's inside of the mouth and then could potentially migrate through the mucosal barriers and then cause an infection, right? So, but if you neutralize that virus by washing your hands or by using hand sanitizers, those kinds of things, those are going to disrupt the shell, if you will, of the virus. So the idea there is to augment that surface barrier of the skin. In terms of the mucous membranes, so those line our respiratory tract, so they're inside of your nose and throat, so inside of your nose. Again, and we talked about this a little bit previously, but the back of your nose, the back of your mouth, and then your throat, those are all part of something called the pharynx. So you got the nasopharynx, the oropharynx, and the laryngopharynx, but basically those are the back of your nose, the back of your mouth, and then down into your throat. So the nose, the pharynx, the sinus cavities, all those are lined by mucous membranes, as is the mouth, as is the esophagus, stomach, intestine, etc. And so the mucous membranes also form a barrier to keep pathogens out. So that's our first line of defense, or the barriers that we have. Basically the epithelia. So the epithelia that forms the skin to keep pathogens out, the epithelia that forms the mucous membranes to help keep pathogens out there. But of course, those defenses aren't entirely impermeable, so things do get past that first line of defense. And so then once they do, then we have a second line of defense. Once that bacteria, let's say, has gotten into the bloodstream, we have a second line of defense to attack it. And so that second line of defense is non-specific defense in that the cells that are part of it basically recognize things that are foreign to the body, things that are non-self. So you've got where it says they're phagocytes there. So phagocytes include 2 types of white blood cells. So you've got neutrophils and then the macrophages. And so the neutrophils and the macrophages are your phagocytes. And so again, what that means is those types of cells kill other things, other cells, for example, or can kill viruses by just enveloping them, by eating them, right? But in addition to those, we also have natural killer cells. So again, natural killer cells, same concept. They're really good at identifying things that are non-self or things that are out of the ordinary, because natural killer cells also neutralize cancer cells in the body. So originally those were self, but because of a genetic mutation, they've become a different type of cell, a cell that's going to engage in unchecked, essentially unlimited cell reproduction. And so that changes the membrane of that cell. It changes the glycoproteins, the sugar proteins that it presents, and then that can alert the natural killer cells, which will go through and actually kill them. And then you've got inflammation. So inflammation is this very non-specific, basically alert for the body. You see certain things with inflammation. You see vasodilation, so blood vessels opening up. You see increased capillary permeability, allows the white blood cells to move out of the bloodstream. You see things like fever, which you can see a little bit further down. That's related to inflammation. It's part of that same chemical messenger cascade. We also make antimicrobial proteins. We make those actually both. We have some antimicrobial proteins here as well, but even once things get inside of the body, we've also got some antimicrobial proteins. So that first line of defense then is keeping pathogens out. Second line of defense is, crap, pathogens are in, now what? And so we got to attack them. But again, that second line of defense basically recognizes things as foreign and just attacks anything foreign. Our third line of defense, which is our adaptive defenses, attack things that we've seen before or things that our immune system recognizes. So those are our lymphocytes. Those are our B cells and our T cells. And it's actually the T cells that do the attacking. But basically, once the T cells are able to recognize a particular antigen, again, an antigen's a pathogen. So once the T cells are able to recognize a particular antigen, they can go to work attacking it. But they have to be able to recognize it first. And so one of the things from the adaptive side of the immune system is it has to be introduced to a particular pathogen. And so one of the ways that textbooks usually describe those adaptive cells, those B cells and T cells, is that they are, in a sense, blind. They can't see unless they can't see a pathogen, more specifically, unless they are presented with it in a very specific context. But once they become really effective at killing that specific pathogen. So we keep the virus out, well, keep pathogens out. If pathogens get inside, we can attack them sort of indiscriminately. And then once they're inside, we also have a discriminative aspect of the immune system that attacks that specific pathogen. So those are the different branches of the immune system. All right, so now we're going to cover each as you would expect. So in terms of the innate defenses, so as mentioned, the skin and the mucosa, or the mucous membranes, either way, act as a physical barrier to keep the pathogens out. So they do that by not only being a physical barrier, but they also have some protective chemicals that they generate. So for example, your skin is slightly acidic. I think in one source I found it was like a pH of, so if 7 is neutral, it's a pH of like 3 to 5 for your skin surface. And so why that matters, though, is because that slight acidity inhibits the reproduction of bacteria. So if you get bacteria on your skin, which we've all got, bacteria on our skin, we tamp down or we limit the reproductive rate of that bacteria through our slightly acidic skin environment. We also have enzymes that we secrete. So a particularly important enzyme is called lysozyme. And so lysozyme is found in saliva, but also in mucus that we secrete in the respiratory tract and then in the digestive tract. Lysozyme is also found in tears, and it's also found in sweat. And so lysozyme is one that actively kills bacteria. So you've got enzymes that you secrete onto the skin surface through your tears and that line the respiratory and digestive tracts that actively kill viruses. And bacteria. This one's just bacteria, so it actually actively kills bacteria. I want to be correct there. Mucin. So we've got mucus that lines, again, the inside of the respiratory tract and the digestive tract. And so that mucus traps a variety of pathogens. So it traps, dust, which isn't necessarily a pathogen, could be an allergen, but it traps things like dust, viruses, bacteria, all those things in that mucus that is on the surface of the membranes inside of the sinus cavity, for example. And then the cells in that cavity are ciliated. And so hopefully you'll remember that cilia are sort of whip-like extensions that are on the surface of a cell membrane that's facing into a body cavity. And so cilia beat constantly, so they kind of just flop over. So they're these extensions from the cell membrane that just flop over. And what that does is it keeps the mucus moving. And specifically, it keeps the mucus moving, if we're talking about the respiratory tract, it keeps that mucus moving down the throat, or in the case of actually farther down the respiratory tract, it actually moves it up the trachea. But either way, what we're going to do is to sweep that mucus and all the things that are trapped in it toward our esophagus, and then we're going to swallow it, which is super gross, right? But what that does is that moves those viruses, bacteria, etc., into the esophagus, which goes down into the stomach, where then we're going to use the acid of the stomach to kill whatever's in there. So the stomach, really high acid environment, I forget what the pH is, I think it's somewhere between 1 and 2 on the acidity of the stomach. So really acidic, it's hard for anything to survive that environment, and so we trap things in mucus, and then we once they're trapped, move them into the stomach, and then we kill them there with the acid. And then we also generate defensins. And so what defensins are antimicrobial proteins. They control both bacterial and fungal reproduction and growth. And defensins are found, they're actually part of both on the skin surface, We secrete some defensins, but they're also part of that innate immune system as well. And then in terms of the other chemicals, so the oil that we secrete, remember that's called sebum. And so so anyway, some of the oil that we secrete is going to be toxic to bacteria as well. So that falls under the other chemicals. So a lot of the things on the skin surface, so we secrete oils, we secrete weak acids, we secrete lysozymes, so all those things then are going to be damaging to the bacteria and funguses that live on our skin surface that would be problematic if we let them regenerate out of control. And then there's more about each of those. All right, so if something then makes it past the skin, makes it past the mucous membranes, well, now we have to attack it. So I mentioned a minute ago that part of our second line of defense are our phagocytic cells. And again, the two sets of phagocytic cells, we've got our neutrophils and our macrophages. So if you think back to the blood chapter, I didn't put a ton of emphasis on it. I talked about white blood cells broadly. I talked about granulocytes and agranulocytes. But one of the things that's in the book, one of the mnemonics, is for the different types of white blood cells and then their prevalence in the bloodstream. And so the acronym that they used, the mnemonic that they used, is never let monkeys eat bananas. So never, neutrophils. The L is the lymphocytes. M is macrophages. And then the E is eosinophils, and then B are basophils. So why that matters for now, neutrophils make up the vast majority of white blood cells circulating in the body. They make up somewhere around 70% of white blood cells. So we got lots and lots of neutrophils, which again are they're these non-specific macrophages that anything that they recognize as non-self, they're going to attack. And so one of the ways that they do that, you can see the pattern recognition receptors. And so pattern recognition receptors are things in the membrane of the phagocytes, so things in the membrane of the neutrophils and of the macrophages that are able to recognize a particular class of thing as being a bacteria or as being a fungus. And so the toll-like receptors are a particular type of pattern recognition receptors. And so the gist of it is, if a macrophage is able to, from the receptors on its membrane, link up with the receptors on the membrane of a bacteria, it's going to recognize, hey, wait a minute, the pattern that's presented by this particular cell that I've just come into contact with, that's not one of our cells. That's a bacterial cell. And so based on that recognition of, oh, this is a bacteria, the macrophage will then hook on to that cell and basically start to pull that cell inside of itself. And so that's kind of what's going on here. So you've got your phagocytic cell that has recognized, hey, this is a bacteria. So it has adhered to that. It's hooked up. So its receptor has hooked up with the receptor on the surface of the bacteria. It pulls that bacteria inside of itself into a phagosome, and then it's going to combine that little vesicle here, the phagosome, with its lysosome. Remember, the lysosome is like the cell's stomach, and so it's a really highly acidic environment. And so basically what it's going to do is to move that bacteria inside of its own stomach, and then it's going to digest it into little protein fragments and whatever else is left, and then it's going to excrete those. So in order for the neutrophils and the macrophages to be able to do that, to undergo phagocytosis, they have to be able to hook onto whatever that invading pathogen is. So they have to recognize that specific receptor. Their receptor has to hook onto the receptor of the invading pathogen, and then they can start that phagocytic process. In terms of the lysosomes, we talked about that. That's the cell stomach. In terms of the helper T cells, trying to remember what I wanted to say about that. So one of the things about particular types of pathogens is that sometimes this whole digestion process in the lysosomes doesn't work. So there are certain things that are resistant to that digestion process. So an example of that is the bacteria that causes tuberculosis. So it essentially inhibits this process, actually one step down. It inhibits this process, it inhibits the joining of the phagosome with the lysosome. And so one of the things that the helper T cells, which are a type of lymphocyte, one of the things that they can do is from the inflammatory chemicals that they create from the chemical, or the, we'll go with chemical mediators that they secrete. So they can secrete something called cytokines, which are these sort of chemical messengers. And so one of the things that the helper T cells can do then is by the chemical messengers that they secrete, they can help the macrophages to generate additional chemicals that include hydrogen peroxide and a substance that's pretty similar to bleach, and then that causes the respiratory burst here. And then that respiratory burst, whether it's the hydrogen peroxide or the substance that's similar to bleach that the macrophage has now created with the help of the helper T cell, that is able to disrupt the membrane of those cells and to kill them. As mentioned earlier, antibodies. So again, antibodies are secreted by a different type of lymphocyte, the B cells. And so the antibodies, one of the things they do in this context is they mark an invading pathogen so that it's easier for the macrophages to see them. So for example, certain types of bacteria, so the bacteria that causes pneumonia in particular, have capsules around their membrane or partial capsules around them. And so that keeps the macrophages from binding to them because their receptor sites are covered up. And so one of the things that the antibodies can do is to basically hook on and then provide a handle that the macrophage can grab onto to then envelop or ingest that invading bacteria in that case. So the antibodies, again, in addition to marking this as, hey, this is something you need to attack, they also provide a physical handle, so they have a receptor site that the macrophages can bind to be able to attack that particular pathogen. All right, so again, the phagocytic cells include the neutrophils and the macrophages. And then our natural killer cells here, so they're a little bit different. One of the ways that they're different is that they are not phagocytic. So they, broadly speaking, the two other ways other than phagocytosis that our white blood cells can kill invading pathogens, basically they can insert something into that pathogen's membrane. There's different names for them. Perforins is one of the names, or perforins, I don't know exactly how it's pronounced. But, you know, like perforation. They perforate the membrane, so they're perforins, I'll call them. But the gist of it is that what they can do is insert these protein complexes into the membrane of a bacteria, for example. And so what that does is creates kind of a porthole in the side of that bacteria, or a porthole in the membrane of that bacteria. And so if we've created this hole in the bacteria's membrane, well, we've disrupted its ability to be selectively permeable. We've disrupted its ability to keep water out, for example. And so by creating those holes in the cell's membrane, the bacteria's membrane in that case, it allows fluid to rush into the bacteria that causes it to lyse. So that causes it to explode, basically, which is pretty cool. So that's one of the things one of the ways that lymphocytes can kill invading pathogens other than phagocytosis. Another way that they can do it, which is related to apoptosis, is that they can fuse granules. So their vesicles can fuse with the bacteria, let's say. We'll stick with that example. And then when that happens, that bacteria, or that vesicle that fuses with the bacteria is going to contain little granules of enzymes that can digest proteins. And so once the granules get inside of the bacteria, they degrade the proteins from the inside of the bacteria. So the result is basically that the bacteria is being digested from the inside, which is also cool. And so then that induces cell death. That's that apoptosis process there. So natural killer cells then kill a variety of things. So as you can see there, they attack cancer cells, but they also attack our own cells that are infected with viruses. So what ends up happening is that either our cancer cells or our own cells that are infected with viruses, as part of that process, so remember that when a virus attacks a cell, it basically hijacks the protein building machinery inside of that cell. And the virus forces the cell, by giving it a different set of instructions, it forces the cell to make a bunch of copies of the virus. And so then eventually that cell explodes, all those copies of the virus get ejected out, and then they go attach onto different cells. And so that's how the viral infection process works. So in that intermediate time where the virus has moved inside of one of our epithelial cells, for example, and it's forcing it to make copies of itself, the membrane of that infected cell is going to be a little bit different. So it wouldn't be what that cell would be like without being infected. And so the natural killer cells, as they move around, are able to bind to cell membranes, and they're able to see this is an epithelial cell, but something's not quite right here. So by detecting that abnormality, the natural killer cells know that they need to attack that particular virus-infected cell. And again, cancer cells, even though they were originally ours, because there's a change in, or a mutation is that change, in the DNA of that cell, one of the other things that tends to happen is you tend to get a change in the glycoproteins on the membrane of that cell. And so that cell, even though it is self, it becomes self but different. And so the natural killer cells are able to pick up on that. And then inflammation, as you can see, and we've talked about this some before. I think we talked about it with the tissue chapter. We talked about the basic steps in the inflammatory process. And so inflammation is, as you can see, this kind of non-specific response to injury. So sticking with the example of mechanical injury, because I think that's probably the easiest to conceptualize. If you get a scrape, for example, in response to that mechanical injury, in that you've deformed the epithelium, you've probably torn some underlying collagen and connective tissue, and maybe you've nicked a couple of blood vessels. So in response to that mechanical damage, you're going to get the release of a host of inflammatory chemicals. And so those are these here. So histamine is in mast cells. And so remember, mast cells cluster really close to blood vessels. And so if there is nearby cell damage, mast cells are going to release histamine. All of these things basically do the same set of things. They cause a combination of vasodilation, they increase capillary permeability, and they increase pain. So histamine is one that does these two for sure: increase vasodilation and increase capillary permeability. So that's going to open up blood vessels so that we can get more white blood cells into the area to help attack. If you've got a cut, maybe some bacteria came in with it. And so we're going to need some white blood cells to attack that invading bacteria, but we're also going to need some macrophages to clean up any of the damaged collagen or cellular debris that's hanging around as a result of the mechanical trauma. So histamine is going to cause vasodilation, open up blood vessels, and then it's going to increase capillary permeability. So remember that the endothelium, the inner lining of a blood vessel, in our continuous capillaries, to try to string this all together as efficiently as possible, in our continuous capillaries, which is what we're going to see in the skin and in skeletal muscle, those endothelial cells have tight junctions that adjoin them. Well, in response to chemicals like histamine, the different kinins, and prostaglandins, that is going to allow those junctions to relax a little bit. And so that's going to create some gaps that our white blood cells can move out of the bloodstream and into that, into the basement membrane and into that loose connective tissue that's going to surround that blood vessel and also be underneath the skin. So kinins do the same kind of thing. They also cause vasodilation, capillary permeability. The difference is, so histamine is generally inside of your mast cells. It's other cells release it too, but it's inside of the mast cells primarily. Kinins are a plasma protein, so they're floating around in the plasma, and in response to other chemical messengers, they initiate this inflammatory cascade that causes increased vasodilation and increased capillary permeability. Prostaglandins, on the other hand, are molecules that are inside of cell membranes, and so if that cell membrane is disrupted, so thinking back to the skin chapter when we talked about the keratinocytes, the skin cells, if you disrupt their cell membranes, that then is going to cause the release of these prostaglandins, and then that is going to be a chemical signal that there's been some cell damage that's happened. And so the end result there are these, as well as pain. So prostaglandins are ones that are really good at allowing for swelling because of that increased vasodilation and increased capillary permeability. But they also hypersensitize the free nerve endings, and so they play an important role in sensitizing the area, making it really painful, because we want to avoid, re-injury of the area, so it becomes fairly painful. Not to mention, as we've discussed previously, that swelling, that fluid that accumulates is mechanical or ends up mechanically compressing the free nerve endings, which then causes pain. And incidentally, any of the non-steroidal anti-inflammatory drugs that you take, so things like ibuprofen, naproxen sodium, aspirin, those are kind of the big three that most people are familiar with. Any of those inhibits the production of prostaglandins, and so by inhibiting the production of these, that's able to decrease vasodilation, decrease capillary permeability, and so those are really good at reducing swelling. And so that's one of the reasons, like if somebody has prolonged swelling, let's say in and around their knee, for example, maybe they got some patellofemoral tracking problems and they got some swelling. And so if they go and take ibuprofen for several days, or they go and take naproxen sodium, which a trade name is Aleve for that, if they take that for several days, usually you're going to see a reduction in swelling, in part because of the inhibition of actually I think primarily because of the inhibition of the production of prostaglandins. So in addition to that, those inflammatory chemicals also serve to mobilize our phagocytes. So again, our neutrophils and our macrophages. So those are down there. The other term you'll see related to the macrophages are monocytes. So monocytes are kind of immature macrophages. So the monocytes are the ones that actually circulate in the blood, but when they're ready for action, they become macrophages. So another way to think about it is we talked about blasts and cytes, so immature cells versus mature cells. So the monocytes are kind of that immature macrophage, if you will. They're not quite ready for action yet. So all of these chemicals then, histamine, the different kinins, and there's a bunch of them, and then prostaglandins kind of start this process in action. So the process is leukocytosis. What that means is allowing the migration of white blood cells from the red marrow, because remember that's where we make our blood cells, is in the red marrow, so inside of the bones, particularly the flat bones. So leukocytosis is the migration of the white blood cells from the marrow into the bloodstream. And then once they're in the bloodstream, those chemical mediators also cause the endothelial cells, again the cells lining the inside of the blood vessels, cause a change in their cellular membrane as well. And so the endothelial cells near the damaged area are going to sprout cell adhesion molecules. And the cell adhesion molecules are basically kind of like handles that stick out from the endothelium that say, this is the place, this is where you're going. And so the white blood cells, as they're floating by, will stick to those cell adhesion molecules that have sprouted from the endothelial cells. So now they're where they need to be. And then they'll engage in diapedesis. So diapedesis is moving from the bloodstream into that underlying connective tissue, or into that, let's stick with that. So into the underlying connective tissue. So they're leaving the bloodstream. And so the way that those white blood cells leave the bloodstream is through that increased capillary permeability. That's again going to open up junctions, going to open up spaces between those endothelial cells, and that'll allow the white blood cells to kind of squeeze through those gaps. And then chemotaxis is once the white blood cells are in the affected area, they are then going to start releasing their own chemical messengers, right? So they'll start to release, where is it going with this? They'll start to release their own prostaglandins as well. I think some of them can release histamine, so the basophils for sure can. So they'll release their own chemical mediators that then attract even more white blood cells to the area. So it sort of starts that positive feedback loop where the initial chemical mediators attracted the white blood cells, and then once the white blood cells get there, they release more of the chemical mediators, and so that causes even more white blood cells to migrate to the area. That's what chemotaxis is. All right. So sorry, had to get a drink and then I messed up. So in terms of the antimicrobial proteins there's two to draw your attention to. The first, there are interferons. So the interferons are generated by virus-infected cells, and that's what you've got going on the graphic there on the right. So you got this virus that's going to invade a cell, so it's able to hook onto the cell, get the cell to take it inside. But as part of that process, the cell realizes, if you will, that something has gone awry. And so that is going to switch on the interferon gene. Remember that genes are the instructions for building proteins. And so then we're going to start to generate, or the cell, the infected cell is going to start to generate some interferons, and then it's going to release them. So what interferons do, there's a couple different things that they can do. They can cause the degradation of the viral RNA. So basically they can cause it to break down, so the virus has a harder time replicating inside of the cell. But they also, interferon is released by the cell, as you can see in the diagram, and it's picked up by nearby cells. And so when the other cells receive that interferon, they know, to give cells more sentience than they have, the cells that receive the interferon know that a neighbor has been infected by a virus, and so they need to take protective actions. And so what that reception of that interferon does is to help the neighbor cell block the reproduction of the RNA of that virus. So if that virus happens to get inside of the neighbor cell, it's able to block replication of that virus's RNA, making it harder for that virus to make a bunch of copies of itself. So basically, the interferons, then the name comes from their ability to interfere with the reproduction of viral RNA. Either they can degrade it directly. They don't attack the virus. This is important because there's one of the questions in the quiz bank is about interferons attacking the viruses. They don't attack the virus directly, but what they do is, remember, the virus or a virus is basically a membrane around RNA. And so if we can keep that RNA from reproducing, or we can make that RNA less stable, then we make it harder for that virus to hijack our cells and turn them into virus-producing factories. So interferons interfere with the ability of viruses to reproduce their RNA inside of our cells. So basically, this cell is probably a goner, but what it tries to do is, in the process, it tries to save as many of its neighbors as it can, even though it's probably too late for this cell. But one of the other things that the interferons do is they signal the natural killer cells. Remember, the natural killer cells are able to detect something, this is either a non-self cell, which is not the case here, or this is a self cell, but there's something not quite right. And so the interferons signal the natural killer cells, hey, I've been infected, come and attack me. Because if we can degrade this cell before it's able to produce lots of virus, we can then slow down the infection process. And so the idea is to, keep our cells from producing, or keep our cells to producing as few copies of the virus as possible. And so if we can attack the virus-infected cells early in this process, then we can keep the virus from replicating, from making too many copies of itself. The other thing is the complement system. And so the complement system, those are plasma proteins. So again, proteins that are floating around in your plasma. And so they are activated by antibodies. And so they, in terms of the name of complement, they complement the adaptive immune system is kind of a way to think about them. And so when those plasma proteins are activated by antibodies, they can do a couple things. The first is that they form membrane attack complexes, or MACs. And so MACs are what I described to you earlier. They are these proteins that insert into the membrane of an invading virus, or let's go with bacteria, because that's usually where we use them. They insert into the membrane of an invading bacteria, and they effectively create a hole in that membrane. And so remember, that can the typical chain of events, I guess, there is that we're going to allow a bunch of water to move inside of that bacteria, which then causes it to lyse or causes it to explode. But in addition to that, the membrane attack, the complement system can act as antibodies in their own right. So by hooking onto an invading pathogen, they can provide a handle that the macrophages and the neutrophils can attach to be able to phagocytize the invading pathogen. So they can act as antibodies in their own right. They can act as handles and as signals that, hey, this is something that you need to attack. All right. All right, so we're through the first two lines of defense, as you can see. So we covered the barriers to pathogens getting inside the body, and then once they're inside, then we're going to try to attack them with a variety of different things, the complement system we just talked about, but also natural killer cells, phagocytic cells, etc. And so in addition to those things that we can throw at an invading bacteria, virus, etc., then we can also engage our third line of defense, which is our adaptive or our specific immune system. So as you can see there's two branches to adaptive immunity. The first one is humoral. And so anything humoral deals with the blood. And so when we talk about humoral immunity, we're talking about immunity that is related to antibody production. And so the B cells, or the B lymphocytes, they mean the same thing. The B cells are going to produce antibodies, which then attach to the invading pathogen and again serve as a signal as well as a handle for the phagocytes, like your neutrophils and your macrophages, to attack those invading pathogens. So that's the humoral branch. It's the production of antibodies. Cellular immunity is carried out by the T cells, or the T lymphocytes. And so in cellular immunity, the T cells, more specifically the cytotoxic T cells, are going to directly attack either a virus-infected cell that's one of our own, or the invading pathogen. So things about it that are different from innate immunity. So it is specific. So the T cells have to see a particular antigen that they recognize in order to be able to attack it. As opposed to, remember, so for example, with the macrophages, they can attack a whole class of bacteria that look the same. It doesn't matter that it's a specific, and I don't know my bacterial classes super well, but it doesn't matter that it's a particular type of Streptococcus bacteria, for example. Versus T cells have to see something really specific in order to be able to attack it. So the innate immunity is much broader. In terms of it being systemic, so the T cells can attack that virus or those virus-infected cells anywhere in the body, as opposed to, like with the inflammatory process, kind of the way I laid it out there a few slides ago, was related to physical trauma. So there was some kind of a scrape or cut that initiated that local inflammatory process, and so you get this localized inflammation, this localized increase in number of leukocytes, of white blood cells. And so that's a very specific response to a specific, potentially bacterial invader there at one site. On the other hand, then the T cells can attack anywhere in the body, other than the brain. And then it has memory. So as you'll see here, when the B cells and the T cells become activated, they make copies of themselves, and one set of copies are your memory cells. And so the memory cells have receptors for a particular antigen. So for example, for the COVID-19 antigen, and they will remember that antigen so that the next time they see it, we can ramp up our defenses much more quickly. Basically, the memory cells function to say, okay, we've seen this pathogen before, we're going to hang around because it's more likely we'll see it again, so we're going to hang around in case we do, and if we do, we've already got a copy of all of these various memory cells, so we can ramp up the production of antibodies and we can ramp up the production of cytotoxic T cells really quickly to mark the invading virus and then to attack the invading virus. So that's where the memory effect comes in. And those memory cells can live for decades. So they are really, really long-lived cells, as opposed to, red blood cells that only last for, I don't know, 3-4 months, as we've talked about previously. Or even the T cells, once the cytotoxic T cells become activated, they only live for about a week after that. So they have fairly short lifespans. Your memory cells, however, have, like I said, a lifespan that can last decades. All right, there we go. All right, so we talked about antigens. So I mentioned, on that first slide, an antigen is something that is going to initiate an immune response. So you can have complete antigens, and so complete antigens include things like viruses and bacteria. So the short thing there is that they, in and of their own right, are going to initiate an immune response. As opposed to a hapten are molecules that don't, in their own right, initiate an immune response, but once they fuse with a protein in the body, then that forms an antigen, and that provokes an immune response. So for example, things like poison ivy, if you're allergic to pet dander, different detergents, those kinds of things, those molecules in and of themselves don't provoke an immune response. But once they're inside the body and then actually bind with our own proteins, then that is seen as a foreign invader, if you will. That becomes an antigen, and so then that provokes that immune response. So in terms of the terminology on there, so for complete antigens, immunogenicity is the ability to stimulate specific lymphocytes to reproduce. So your lymphocytes, again, are your T cells and your B cells. So immunogenicity then means it's going to cause T cells and B cells to proliferate. And then reactivity is the ability to react with those activated lymphocytes. So to react with those T cells and B cells. In terms of the antigenic determinants, so it is the part of the antigens that are immunogenic that are going to actually provoke the response. So they have different receptor shapes, which we've all seen a lot in the last year talking about the coronavirus and the spike proteins on it, because those are what the vaccines actually work on. The vaccines carry instructions for building or parts of the spike proteins, and so those spike proteins then are those antigenic determinants. Those are the things that our antibodies can bind with. So those are the things that are going to actually provoke the immune response. The MHC proteins, so I got to fight my reflex here. When we talk about skeletal muscle, there are different chains of myosin. There's myosin heavy chains and myosin light chains. And so every time I see MHC here, I keep wanting to say myosin heavy chain, which would be accurate if I was talking about muscles, but I'm not. So in the case of the immune system, MHC stands for major histocompatibility complex. Major histocompatibility complex. And the relevance or why those proteins matter is those help to identify things that belong to us. So those are proteins that we use to identify self in our cells. So where that comes in. So cell membranes are going to display different types of MHC proteins depending on which type of cell we're talking about. And so the T cells, for example, are going to use those complexes to identify, okay, this is a cell that belongs to us, or it's not, and if it's not, then maybe that's something that we're going to end up attacking after the T cell becomes activated. All right, so one of the things about both B and T cells is that they have to mature. We talked about this a little bit with the trying to think of the chapter, with the lymphatic chapter. So the B cells mature in the red marrow. The T cells mature in the thymus. So even though both the B and the T cells are generated initially inside of the red marrow, the B cells hang out a little while longer. The T cells are exported to the thymus and they mature there. And so by maturing, what they're doing is they're developing immunocompetence. They're developing their ability to recognize and bind to a specific antigen. So your B and T cells are going to have a wide variety of receptors. And I forget, okay, this is good. I wanted to make sure this was a slide where I had the number on them. So they're going to have this wide variety of receptors on them. And we actually have between all of our different B and T cells, over 1 billion different antigen receptors. And so one of the things that's interesting about that is obviously we don't have 1 billion different genes. We don't have 1 billion different sets of instructions for building proteins. So what happens is while those cells are developing, while they are maturing in the red marrow or in the thymus, they have in their stem cell state, they have basically just bits and pieces of genes for building those surface receptor sites, and then they basically just shuffle those proteins over and over and over again to build, between all the different lymphocytes, to build a whole bunch of different possible combinations of receptor sites. So between all of our billion different combinations of lymphocytes, we have some that are going to be able to recognize the COVID-19 virus, even though they've never seen that virus before, or that's not actually the virus, that's the disease it causes. So they're going to be able to recognize the coronavirus, to use the appropriate terminology. And so once they're able, to be really specific, it's SARS-CoV-2. So even though we've never seen that before, even though that's a new virus that just recently evolved, we still are going to have some lymphocytes that are going to have the receptors for that virus because, again, you've got lots and lots of lymphocytes, and each of them is making a different set of receptors. And so between all those different receptors, if we've got more than 1 billion different combinations of receptor sites, one of them is going to match up closely enough with the spike proteins on the coronavirus that they'll be able to attack it. So that's one of the really cool things about these cells is during that period where they're developing immunocompetence, they are sprouting these surface receptors that can bind to viruses that or receptors that don't even exist yet, which is pretty cool. So that's developing immunocompetence, the ability to recognize and bind to a specific antigen. And so once they are immunocompetent, so once our T cells have matured, then they can leave the thymus and they will then be transported to or end up in places like inside of lymph nodes or inside of the spleen. And so before they actually come into contact with that particular antigen, so think of it as they are the lock and the virus that they match up with would be the key. Before they meet up with that key, they just kind of hang out. They're just waiting to encounter that specific key. That may never happen, right? So if you've got 1 billion different combinations, you're not going to encounter 1 billion different viruses. And so some of those T cells never get to do anything. And it's the same thing with some of the B cells. So some of them just hang out forever. And the ones that haven't actually yet matched up with their specific antigen, those cells are called naive. But once they have met up with the antigen, then they are no longer naive. So you see that sometimes actually related to COVID. You'll hear that this particular population is naive to the virus, meaning that their lymphocytes in this case have never seen that particular antigen. And then once they have, they're no longer naive and they can ramp up their immune response really quickly. So one of the things with both B and T cells is once they bind with that particular antigen, the cells have to become activated to do anything. And so activation of these cells is a 2-step process. So one of the ways that I like to one of the analogies I like to use here is that it's kind of like if you ever started a car where you have to not only put the key in and turn it, that's step 1, but step 2 is you have to have your foot on the brake in order for the car to actually turn over. And so you got to do that 2-step process to actually start the car, to activate the car, if you will. Combination key turn and brake stomp, or foot on the brake. Similar idea here with the B cells and the T cells. They have to not only bind with the invading or with the antigen, but they also have to receive some kind of a second signal. So they could receive that second signal from something called a helper T cell. They could receive that second signal from what's called an antigen presenting cell. They could receive it from these chemical messengers called cytokines. But either way, there's 2 steps in this process for them to be active. And once they become active, then they make a bunch of copies of themselves. They make a bunch of clones. And then the clones that they make fall into 2 categories. There are effector clones that actually do something, and then there are the memory cells. So, for example, if we activate a B cell, the effector clones are going to be plasma cells, and so the plasma cells are the ones that are actually going to make the antibodies. But then, in addition to making those plasma cells, that B cell is also going to make clones of itself that function as memory cells. And so the memory cells have that same antigen receptor on them. And so we're going to have a bunch of memory cells just kind of hanging out, again, potentially for decades, inside of our lymph nodes and our spleen and circulating. And those memory cells have that same antigen receptor on them, so that when they see that antigen again, so if you encounter the coronavirus this year and then you encounter it again 3 years down the road, you have cells that have the receptors for that spike protein, so they can ramp up your immune response really quickly. In terms of education, I don't really care that you know about this because the book only even talks about T cells anyway. But the gist of it is with education of the lymphocytes. So the T cells, because again, they're the ones that actually go through and will attack the invading pathogen, it's important that the T cells are able to recognize us. Because if they don't, then they're going to start to attack and kill our own cells. And as you can imagine, that's a problem. And so the gist of it is the positive selection part is that the T cells, when they're maturing in the thymus, that they can recognize that this cell belongs to us, and they do it by binding to that major histocompatibility complex, the MHC. So they bind to that MHC, and they're able to, based on the shape of that protein, recognize, okay, this is one of our cells. So that's positive selection, so that they can recognize us, they get to stick around. If they can't, they die. And then we go to negative selection, which is there's going to be a protein, an additional protein embedded, or a fragment of one, embedded in that MHC, in that complex. And so if that protein is one that we normally make, then, and so then that would cause the T cell to attack it. That T cell has to die. If that protein is one, let's stick with this. It can't initiate an attack on a protein we normally make. So it has to be able to recognize us, and then it has to not attack proteins that we normally make. Because if it does attack proteins that we normally make, then it's going to start to attack our own cells. And so what you get there is an autoimmune condition. So an autoimmune condition is one of the topics, or one of the potential topics for this week's discussion board. But an autoimmune condition is where your immune system attacks yourself. So those include a variety of different conditions, from multiple sclerosis to things like celiac disease, Crohn's disease, rheumatoid arthritis, and a number of others. All those are autoimmune conditions. And so what's happening there is there's something wrong with our immune system where the cytotoxic T cells in particular are attacking our own cells and causing them to be damaged, and they're starting this inflammatory cascade. So to prevent that, ideally, our T cells go through this education process where they learn what we look like and not to attack it. All right, antigen-presenting cells. So again, an antigen is that foreign invader that is going to initiate an immune response. And so antigen-presenting cells are able to show the lymphocytes, hey, this is what the invader looks like. That's kind of their job. So one of the primary antigen-presenting cells are our dendritic cells. So dendritic cells we talked about a little bit with the skin. They're in the deeper layers of the skin, but they're also in the mucous membranes. And so what the dendritic cells do is they're able to engulf that invading virus or bacteria, and then they break it down. So they are phagocytic, I believe. But at any rate, they're able to engulf that invading bacteria or virus, and then take a piece of it and then display that piece of it on their outer membrane. So they're presenting the antigen to be received by B cells or T cells, naive B cells or T cells. And so the actual process for that is, let's say that an invading bacteria makes it through the mucosa into that underlying connective tissue, where the dendritic cell grabs it, moves it inside of itself, and then breaks that bacteria down and presents part of that bacteria on its membrane. That dendritic cell then is actually going to migrate into a lymphatic capillary and eventually end up in a lymph node. And we want it to end up in a lymph node because, remember, that's where, for the most part, our B cells and T cells hang out. And so by moving into the lymph nodes, it basically ensures that the B and T cells are going to see this antigen. It's basically running into the room where they all are and going, hey, look what I found, right? And so it's then going to introduce those naive T cells and B cells to this particular antigen, which is step one in that activation process. So your T cells, before they become active, can't attack anything. So first, they got to know they have to be introduced to the antigen, so they have to know what they're looking for. And so after they bind with that antigen on the antigen presenting cell, then they can become active. And then actually the antigen presenting cells can complete step 2 by themselves. So for example, there's a class of T cells called helper T cells. And so if this dendritic cell presents this piece of bacteria, and so in terms of the picture here, this is a macrophage. As I was talking, the picture was distracting me because I realized I hadn't described it. Anyway, the dendritic cells, when they present this antigen to the helper T cells, they actually have a protein that will join with that helper T cell and basically complete step 2 of the activation process. So they can both present the antigen to the helper T cells and activate those helper T cells. And once activated, the helper T cells can release cytokines, so chemical messengers, that can activate other cells, like our B cells. So that's an important thing. So the helper T cells help by activating other cells. So that's one of the things that they can do. And they can become activated themselves, again, by these dendritic cells. They can also become activated by macrophages, as you can see there. And then the B lymphocytes, so again, B lymphocytes are going to be hanging around in our lymph nodes. And so if they are presented with an antigen by the dendritic cells or by the macrophages, they can become activated, or they can bind with the antigen themselves. Like they can actually envelop the antigen themselves, and they can present it, but they don't become active until they receive the cytokines, the chemical messengers from the helper T cells. But once they do, once the B cells and the cytotoxic T cells receive the cytokines from the helper T cells, they are now active. And why that matters is active B cells and cytotoxic T cells can make copies of themselves. So that's why that matters. So any of those cells, B cells or T cells that aren't active, can't make copies of themselves. If they can't make copies of themselves, they're not really doing anything. All right, so that's kind of what I was describing on the last slide. So if we've got our little B cell here, it's going to bind with an antigen. And then we're going to say that this B cell has received a cytokine from our helper T cell, and so now it is active. And so now it's going to proliferate. It's going to make copies of itself. And so the copies of itself have the antigen receptors on them. And so then those copies, those clones, end up as 2 types of cells. So you got your plasma cells here, and then you got your memory cells. And again, remember the memory cells just kind of stick around. They're hanging out in case we see this antigen, this particular invading pathogen again. So that's the job of the memory cells. Like, you guys just hang out over there, wait, in case we see this again, you're going to be in charge of ramping up the immune response next time. But for now, you guys just hang out. Then we got the plasma cells. So the plasma cells, you can see there's a lot more of those. So the plasma cells are going to secrete antibodies. And again, antibodies will bind to that antigen. And we'll talk actually on the next slide about the things that they can do. So one of the things I want you to know, plasma cells secrete antibodies. And the antibodies, if nothing else, are going to mark the invading antigen so that the phagocytic cells will attack it. In terms of the primary versus secondary immune response, got to remember what I wanted to talk about there. Oh yeah, the lag time. So the first time you see the virus, this whole process of making a bunch of copies of the B cells and also the T cells takes several days. So it takes almost a week to ramp up production, to maximize production of these plasma cells the first time. But the second time, so if we see this same virus again 2 years from now, remember these memory cells are going to be hanging out, and if they see that virus, they are going to replicate really quickly. And so we actually, where the first time it took us 7 days to maximize antibody production, the second time we see that same virus or bacteria, it's only going to take us 2 to 3 days to maximize antibody production. And so this is some of why things like vaccines work, is that if you, by introducing the vaccine, you then introduce this particular virus, and in some cases bacteria, but you introduce this particular virus to the body so that it has seen it before, your your lymphocytes have seen this particular virus before, you've created memory cells for it, and then you can ramp up the immune response in half the time that it would have taken if this is the first time you'd seen that virus. So in the context of something like COVID, why that matters is basically once that virus gets inside the body, it's going to start replicating really fast. And so it's going to take, basically a week for our immune system to ramp up and be like, oh crap, we got a bunch of these viruses we have to start attacking. And your immune system is kind of already behind the 8 ball. It's already getting far out replicated by this virus. And so if we can prime the immune system by introducing it to, in the case of most of the COVID vaccines, a fragment of this virus in advance, we've got memory cells hanging out that can then, in half the time, ramp up the production of the antibodies, and then, as you'll see on the T cell side, same concept, we can ramp up the production of the cytotoxic T cells so that the virus doesn't get so far ahead of us in terms of getting a response and trying to attack it. So it basically cuts our response time in half by having seen that virus before. So in terms of immunity, so there's active immunity and passive immunity. So active immunity includes both infection. So as we all know now, once you've gotten infected with we'll stick with COVID, once you've gotten infected with a virus once, you're then going to generate a bunch of antibodies in response to that, and beyond the antibodies, you're going to generate memory cells. And so those memory cells are going to hang out so that once you see that virus again, you'll be able to attack it really rapidly. So by seeing the virus once, we generate memory cells that ramp up a quick immune response. Or we can get around feeling like crap for a week or two, and we can do a vaccine. And so the vaccine does the same thing. It presents either the virus or a fragment of the virus, depending on how the different vaccines work. But that creates those memory cells that direct the secondary immune response. Passive immunity is what we saw with where people were taking plasma from COVID survivors and introducing that plasma to somebody with an infection. And so the idea there is by taking the plasma from somebody who has already gotten through a COVID infection and introducing it to somebody who's currently infected with the virus, what you're trying to do is to give the person who's got a current infection a bunch of antibodies. And those antibodies then are, of course, going to bind to the COVID virus. They're going to give our macrophages and neutrophils something to look for and something to attack. And so the idea is that you maximize the efficiency of some of those phagocytes. You sort of give the immune system a leg up by giving it those antibodies. You basically, without having the delay of our own B cells, taking that week to ramp up antibody production, you just throw a bunch of antibodies from somebody else at it, and then you show the phagocytic cells what they're looking for. That's the idea behind the plasma therapy. One of the downsides to that, though, is because you've just given them a bunch of the antibodies, it doesn't present as much of a challenge to the B cells, so you don't make quite as many of the memory B cells, and so then the secondary infection, or the secondary immune response, is not quite as robust. So that's kind of one of the downsides there. But if you're administering plasma antibodies to somebody, it's probably because they're probably not in a good spot anyway, and so we'll worry about the secondary immune response later. Don't worry about all this stuff. I was following the textbook really closely with this, so don't worry about this. But what I am interested that you know is the things that the antibodies can do. So here's our little antigen, here's our little antibodies. They'll bind to them, and then they create, as you can see, an antigen-antibody complex. And so antibodies can do a variety of different things. I kept saying that they form a sort of handle on the pathogen, which they do. But in addition to that, so by binding to, sticking with COVID, by binding to those spike proteins on the outside of the virus, it keeps that virus from binding to our own epithelial cells, for example. And so by keeping it from binding to those cells, if it can't bind to the cells, it can't get inside. If it can't get inside, it can't make a bunch of copies of itself. And so one of the things that our antibodies do then is they prevent that binding of the virus to our own cells. But other things that they can do is, because those antibodies usually have multiple receptor sites, they can actually attach to multiple different things. or the same antigen, but on multiple different cells or multiple different molecules. So in this case, what you've got red blood cells. This is somebody, the depiction here is that somebody got a wrong blood type transfusion. But let's pretend these are little coronaviruses. And so one particular antibody can bind to multiple copies of the virus, and then that makes this big clump of them. And then the macrophages can basically, they can phagocytize that big clump of viruses. If we're talking about a smaller particle, then they can bind to the smaller particles and they can form this big mesh network of a combination of antibodies and that particular particle. That's called precipitation. And then same concept, the macrophages can come through and eat it up. And then the other thing they can do is they can activate complement. So again, complement are plasma proteins that complement the adaptive immune response. And so one of the ways that those complement proteins, as mentioned, kill cells is they embed in the membranes and they create a hole. So they can activate complement as well. And again, complement can also serve as an antibody in its own right, serving as a sort of handle or a binding site for the macrophages. All right. So those are our B cells. So now on to the T cells. So again, the thing I want you to know about the B cells, so the most important thing to know is that the B cells produce antibodies. So remember, they have to become activated first, and they make copies of themselves. And so the copies, there's two types of copies. And so in terms of the copies, they're going to have, when I say they're copying themselves, they're going to have the same antigen receptor. So when they make copies, they have the plasma cells that make the antibodies, and they have the memory cells that stick around for the next time we see that pathogen. Similar concept with the T cells. So there's two types of T cells. One of the things to not let throw you is the book talks about CD4 cells and CD8 cells. So the two types of T cells, you've got the helper T cells, which again, they help in that they activate the other cells. So they activate the B cells and they activate the cytotoxic T cells. So the CD4 cells are helper T cells. Just think about them as that. It's probably the easiest way to conceptualize them. And then the CD8s, those are our cytotoxic T cells. So the cytotoxic means cell toxic. So the cytotoxic T cells are the ones that actually go through and attack the viruses, or they attack virus-infected cells. But they can't, so your CD8 cells that are floating around, or what will become your cytotoxic T cells, that are floating around in your blood or in your lymph, if the coronavirus floats past them, they're not going to recognize and attack that initially. They're naive to that, right? Or if they do have the particular receptor for it, they'll bind to that antigen, but again, they're not activated yet, so they can't actually do anything. They'll just bind to it, and they'll see that they have a matching keyhole to the key of that particular virus, but they're not active, so they can't do anything about it. So what they have to do then is the CD8 cells, the ones that are going to become the cytotoxic T cells, bind to those major histocompatibility complexes. All of our cells are going to have these class 1s, and so that's where those T cells had to recognize this is a cell that belongs to us. So they're able to recognize that this is one of our cells. And under normal circumstances, an epithelial cell, for example, is going to present not only that MHC complex, but it's also going to have a protein or protein fragment embedded in it, which is going to be one of ours. And so the CD8 cell, which again, when activated, becomes a cytotoxic cell, that CD8 cell will bind to the MHC of one of our own epithelial cells, and it'll say, okay, this is one of ours. And then it'll see the protein embedded in that, and it'll say, okay, this is one of ours too. Don't attack this. But if that particular cell, one of our epithelial cells that's infected with coronavirus, for example, because it's producing a bunch of copies of the viral RNA and it's producing a bunch of copies of the virus itself, embedded in its little MHC complex, which is redundant, but embedded in its little MHC complex, it's going to have a protein that is a non-self protein. And so if that particular T cell has an antigen for it, it'll bind to that and it'll know to attack it if that T cell is activated. And then the class 2 MHCs, those are on our antigen presenting cells. So those are on things like our dendritic cells, on our macrophages, and then on our B cells. So as mentioned, the T cells have to be activated either by the antigen presenting cells or by T helper cells. We'll add that on there as well. And so once those T helper cells, for example, are activated by the antigen presenting cells, then they'll secrete this particular cytokine IL-2, which, as you can see, stimulates proliferation of the T cells and the B cells. So then the T cells will split into, so here's our helper cells. The T cells will split into both cytotoxic T cells and memory cells. So again, the cytotoxic ones are the ones that actually roam around in the blood, and they look for, as soon as they see that specific antigen, if they're activated, they'll actually attach to that cell, and then they'll release the little granules that we talked about earlier, which will then degrade the proteins inside that cell and cause it to die. So if we've got a cytotoxic T cell that is circulating in the blood, so let's say we've got a CD8 cell, one that's going to become cytotoxic, and it's cruising around in your blood, and it docks with an antigen, or it's cruising around in the lymph. It's in the lymph node. That's where we're going to put it. And it docks with an antigen-presenting cell, say a dendritic cell, inside of that lymph node. That CD8 cell now becomes a cytotoxic T cell. So it is an activated T cell that makes a bunch of copies of itself, but in addition to that, it is activated. So as soon as it sees that antigen, as soon as it sees that coronavirus antigen, it's going to bind to the cell that's presenting that antigen, and it's going to kill that cell. So as that now activated cytotoxic cell is roaming our lymph and then eventually roaming our bloodstream, if it sees an endothelial cell, for example, that is presenting a coronavirus antigen on its surface, that cytotoxic T cell then is going to bind to that cell, release its little granules, and then it's going to kill that endothelial cell. So that's the way that those cytotoxic cells work. The brakes on this whole process are our regulatory T cells. So rather than releasing excitatory cytokines like IL-2 that cause T cells and B cells to split and make a bunch of copies of themselves, the regulatory T cells inhibit that. So they tamp down the immune response. And so they serve an important function of not letting our immune system sort of spin out of control and just attack everything. And so in that, they help prevent autoimmune responses. Let's see. We talked about the helper cells quite a bit. So here's a helper cell activating a B cell. Here's a little, there's an antigen presenting cell, a helper T cell secreting its IL-2, and then activating what will become our cytotoxic cell. All right, so there's the cells involved in the adaptive immune response there. And then I was going to show you, but I'll skip it for now. So also on Canvas, there is a graphic that comes from the textbook. And so it basically has like all the different immune defenses and kind of the chain of events that happens in each. So that's posted in the immune system module just as a PDF. So make sure you check that out before you do your discussion board this week. So like I said, discussion board this week, you got 2 options. So check those out and do whichever one is more interesting or useful for you. And I look forward to reading your responses, and we'll see you next week.